life cycles and emergence of multicellularity. So for you to have an expectation of what will happen in the next few minutes. So today I would like to share with you our work on the evolution of irreversible somatic differentiation. And in a broader context, this is an episode in the evolution of multicellularity. Like billion years ago, our ancestors were unicellular. So every single cell was an independent organism. It could sustain itself. It fulfilled all the necessary tasks by itself. But the time passed and evolution takes place. And now we find quite a number of species having multicellular phenotype. So our bodies are made of cells, and every cell is not an independent entity anymore, but just a functional unit of it. And how cells give up their independence and their Darwinian properties is one of the most challenging issues. It's one of the most challenging issues in the evolutionary biology. Fortunately, evolution of multicellularity happened multiple times. And what you see are 7 photos with 7 organisms whose multicellularity emerged independently. So we can compare between them and study the differences and similarities. And if we just heuristically sort them by complexity, it can be done by multiple ways, but I hope you will be on board with me. We find that like this first and the simplest multicellular organism are just a clump of cells that prefer to stay together. But if we go for more complex ones, we find that cells begin to become different. And it starts with maybe 2 different cell types, as you see on the figures in the middle. And if we go for larger, like macroscopic multicellular organism, we find hundreds and thousands types of cells that perform all the different functions. I'm interested in the very, very first steps of this process. Well, okay, the later steps are also very interesting, but I cannot really do much about them right now. So I start with the very beginning. And what we see at the very first steps of complexity evolution is that the earliest specializations are the specializations into somatic and vegetative functions. For example, in cyanobacteria, we have heterocysts, and those are the cells that are specialized in nitrogen fixation. It can only be done in the absence of oxygen. So for a photosynthetic species, it's a tricky task. So what cyanobacteria heterocysts do, they encapsulate themselves within several layers of the cell wall. And then in anaerobic conditions, they fixate nitrogen for the entire colony. But then they give up access to oxygen and suffer in their fitness a lot, maximally. Another example, totally different, is in terms of position on the tree of life different, is a Pleodarina. Those are green algae. And what you see is a colony that's made of half and half of big and small cells. And the big cells, they will grow and develop into new colonies eventually, but the small cells will not. They have flagella and they move the colony around and keep it to the top of the water surface, so the colony can photosynthesize. And the trade-off here is that both cell division and motility, they use centrioles. And since the cell have only one set of 2 centrioles, those tasks cannot be performed simultaneously. So half of the cell go for one task and another half of the cell goes for another task. It's important to know that those organisms that you see, they didn't invent those tasks. They existed before. So we can consider more simple and sometimes more complex relatives of those species. And they also do the same, but they do it differently. They distribute tasks in space. Not in space, sorry, but in time. For example, there are filamentous cyanobacteria that don't develop any heterocysts. They still fixate nitrogen, but they do it during nighttime when there is no light and photosynthesis cannot be performed anyway. And there is an entire range of different relatives of Pandarina. So this one on the left is gonium. The cells look almost the same, but they're all identical, and the organism is just 16 cell size. So what do? Is again, they move during the daytime and photosynthesize at the next to the surface of the water, but at the night time it slowly sinks, but it reproduces there. And if we go all the way to the most complex relative in this lineage, it's Volvox made of thousands of cells. It has a structure, and germ cells are inside this spherical body, and somatic cells are outside. So even more complexity can be developed from this intermediary form in the middle. And if we think of it from evolutionary perspective, if we see that temporary specialization is already available for ancestors of those organisms, how it is possible for the permanent somatic differentiation to evolve? Because, for example, consider yourself as a single cell in an organism. Differentiate into soma, it's probably the worst possible scenario you can get, because you simply abandon your long-term fitness. It's just zero. You don't produce, maybe you divide, but your offspring will not be part of the next generations of the organism. And whatever benefit you bring to the colony, it doesn't pay you personally back. And for the organism perspective, like before, if you have a temporal differentiation, like you do something for some time and then return back to the basic shape and you can divide and reproduce, you now suddenly develop some cells that will certainly die, and it's essentially in a sense it's a waste of biomass, because you forbid part of yourself to reproduce. So like, what if those somatic cells that comprise our bodies at the very end of our life just try to produce offspring? In a very basic way, it's a very meaningful strategy. Because, okay, your life cycle is over, now you want to contribute more to the, as much as possible to the next generation, and you use all of your cells. But no, you don't. Well, most of us will die eventually, unfortunately.
Okay. Can I ask you a question? Sorry.
Yes, please.
Yeah, so this notion of permanent somatic differentiation, like with all these like new experimental techniques, we're seeing that these terminally differentiated cell types can become de-differentiated in different like contexts, like wound healing and things like that. So what do you mean by permanent? Because they clearly retain this capacity to change their type.
Yes, very good question. Here I do, what the differentiation that I consider is not a terminal one. Like the terminal, I mean, you come to the state and you don't divide anymore. No, I assume that somatic cells can divide eventually, but what they don't do is that they don't contribute to the next generation of the organism. My arm will not become my child.
Okay.
So that's the way, that's the phenomenon that I try to investigate.
So distinguishing between cells which are clearly somatic and cells which are gametes, I guess. Is that what you mean?
Gametes is about sexual reproduction. I'm doing asexual life cycles.
I see, yeah, okay.
But otherwise, yes. So it's like ability or inability to give the organism level offspring.
Got it.
So in this context, we can talk about three classes of development strategies. On the one hand, we may have a situation where no differentiation happens and cells don't really specialize into any specific function. We can think about reversible differentiation, where cells do specialize, but temporarily they can revert back. Not necessarily they will do it, but at least they have an opportunity to do so. And finally, the developmental strategies is irreversible differentiation, where cells specialize into somatic task and they stay like it, and their offspring, other cells, will be of the same function. And that's what we observe in animal species typically. So we're interested in what are the conditions that makes reversible somatic differentiation to be the evolutionary optimal strategy when it evolves. What do we need for that to happen? Because like in plants, it doesn't happen. In plants, well, the fruit develops from cells that were previously just well, they were part of the branch of the. They were not destined to become germ cells, but sometimes it happens like in us. So we built a model, and the model is about some simple multicellularity because the organisms in our focus of attention are small and not very complicated, and it's a population of multicellular organisms with different cell types. And here the tricky part is not really to put irreversible differentiation to the model, but to put the alternative, the reversible differentiation, because we need to consider cells that become specialized and then de-differentiate back if you wish to, or differentiate into germ function. So we consider a life cycle. And the life cycle, it starts with always in a single cell in our model, and cell divides simultaneously all the time. So we have a specific generation of cells, and at generation n, we have 2 to the power of n cells in the organism. But it's not just the cell division, what happens in the life cycle. We also have cell differentiation. We consider that the organism contains 2 different cell roles. It's not cell type, it's something temporary. So it's a role of the cell. It could be a germ role. Germ. So the germ role cell can give rise to a new generation of organism, but it doesn't provide any advantage to the organism growth. It doesn't really help organism to be better, faster, and stronger. On the other hand, we have soma role cells. And soma role cells, they do provide such an advantage. And having more soma role cells is very beneficial for an organism. But unfortunately, those cells, they cannot give rise to new generations. They don't participate in organism level reproduction. So they change their roles, and we tie the change of the roles to the events of the cell divisions. So at every time cell divides, it's 2 new cells. They can be the same, or they can be different, and maybe they both could be of the opposite role. And in total, we have, as you see at the bottom left, 3 opportunities for each cell role, and each opportunity can occur with a certain probability. And those 6 probabilities, they constitute the developmental strategy. So I hope this figure is, well, it speaks for itself. If not, please ask me, I will explain it, because it's pretty important for that context. Okay, fair enough. On the 3 types of the strategies that we had before, the no differentiation, it's where no differentiation happens. Germ always goes into germ. And the strategy is like the first element, germ goes to 2 germs, is always 1. 2 remaining strategies for germ differentiation is 0, and the remaining is relevant because soma never appears. Irreversible differentiation is a situation where you can go from germ to soma role, but once you are in the soma role, your offspring will always stay in the soma role. So the first element is less than 1, so there is an opportunity for differentiation from germ to soma, but there is no opportunity to differentiate back, so soma to double soma is always happens with a probability of 1. And finally, the rest are the reversible differentiation strategies. It's the most space of differentiation strategies are reversible ones. And what we need here is to have the very first and very last element to be smaller than 1, so they can switch back and forth. What do soma role cells do? So they speed up the process of growth. If you have more cells, you will divide faster. And at each time step, the fraction of soma roles in the organism is computed, and based on this fraction, the duration of that division step is computed according to a formula. So we choose this function. It's a sort of sigmoid-like function when soma role cells don't contribute anything if they are smaller numbers, and then the division becomes faster. With more soma cells you have, the faster it is. And once it exceeds the saturation threshold value, no additional soma cells brings anything. We choose this function not because we know something about soma, but because we just don't know how they could contribute. And with this functional form, depending on the parameters, you could have multiple different patterns. You could have the constant if the maximal benefit is 0, so it's always be the same. You might have a linear function, non-linear function, step function, or something in between all those. Very flexible functional form. And you want to know which of those patterns really matters. So at the end of life cycle, there is a reproduction and death. So once the organism reaches the maturity size, then it just dissolves, and all somaroles that happen to be in the somarole at the end of life cycle, they die, and every germ role becomes a new organism, and it starts the growth in you. So overall, in terms of optimization of the life cycle, what's necessary is that you want to have as much germ role cells at the end of life cycle to have a lot of offspring, but you want to have as much somarole cells during the life cycle to complete it as fast as possible. Of course, you cannot satisfy both. You cannot maximize both independently, and therefore you have to go for some trade-offs and find the optimal way to distribute your biomass between germ and soma. And maybe some of those optimal strategies are irreversible, like we see in a number of complex organisms. So the overall pipeline of the research is that of the project is that once we know developmental strategy, we know what the number of offspring will be at the end of the day. If we know what was the somarole benefits, this profile, we know how long will it take. If we know both, we can compute the population growth rate. So this is Euler-Lotka equation from demography field, and we have entire paper dedicated to the fact that we can use it for those systems that we study here. And please know the sum. What happens in here is that our developmental strategy is stochastic. So those differentiations that happens with different probabilities, and different organisms, they will develop in different compositions eventually. So the population as a whole contains a very diverse range of the organism, even if they share the same developmental strategy. So we need to run multiple trajectories of development, and we average over all of those to find the population growth rate is lambda. And I mean, since the organism multiplies, the whole population will grow, and it will grow exponentially. And this population growth rate is how fast is population growth in the system. Once we know the population growth rate, what we can do, we can just compare it across different developmental strategies and find the best one, because it's a population developmental strategy that gives the largest benefit in terms of selection. And then we can check what is developmental strategy that is evolutionary optimal, and check if it is a irreversible strategy or not. And then we can screen through control parameters and find what combination of control parameters promote evolution of reversible differentiation. And that's what we did, and what I will show you for the rest of the work, almost. So the results, and I will start with the most significant finding, at least in theoretical context, that shaped our thinking about evolution of differentiation strategies a lot. And it's the fact that the model that I have shown you does not allow irreversible somatic differentiation to evolve at all. So we first run many simulations, and then we start to think. And yes, thinking helps. So what happens? In the setup that I just explained to you, for any reversible strategy, there is a reversible counterpart that provides the same number of offspring, but does it faster. In reversible strategies, what happens? The number of somarole cells, it cannot goes down, because germ goes into soma, but somas always stays the same. So the fraction of somarole cells, it only increases in time. And it increases exponentially, well, like as a saturating exponent with the number of cell division. It approaches 1. And for reversible strategy, so once you have any irreversible strategy, the green one, once you know that at size, or it's after 12 division, it must fragment, you know how many somaral cells there should be, or how many germinal cells should be. You can find the reversible strategy that achieves the same fraction, but within a single step, the expected fraction. So you just jump into that point and then stay the same in average. So you have the same number of offspring, but during your life cycle, you have much more somaral cells. You just develop much faster. But those strategies, the reversible strategies, they rely on dynamic equilibrium between germ and soma. A lot of germ and soma cells constantly transform into each other, so they keep the expected number to be the same, the expected fraction to be the same across the life cycle. And we cannot move forward without penalizing those high differentiation rate strategies in any way. But how it can be done meaningfully? So return to the model, and we introduce differentiation costs. So like, if you want to change the type of the offspring, you need to invest additionally. You need to remove some proteins, you need to introduce new proteins, you need to rewire your regulation network. There should be quite some processes that take part in differentiation, comparing with just cell division. So what we do now is we assume that the doubling time from generation n to n+1, a generation of cells, so one doubling time, is a product of two factors. One is a composition benefit that I discussed above, this sigmoid-like function, and another one is differentiation cost. And for every cell differentiating, there is a cost. And if both of your offspring are of different roles, then it's double of cost. And we consume this differentiation as 1 plus average costs of cell differentiation across all cells in the organism. So if you don't differentiate, that's the model that we had before. And if you do differentiate, then the more differentiation events happens, the more slowly it takes. And that's when we got our reversible differentiation. But again, a little bit of overview of the model, because it's quite complicated. I have to admit it. We should be simpler, but we have a set of evolvable parameters. That's our developmental strategies. 6 numbers. I will not show it to you, but I will show the classes. No differentiation, irreversible, or reversible differentiation. And this is all controlled by a number of control parameters. First, we have differentiation costs, 2 of them, because differentiation in one direction might be different from differentiation into another one. We have a maturity size when the fragmentation happens. And we have 4 parameters describing our somaral benefits. So, and we want to know under which combination of those totally 7 parameters reversible differentiation evolves. And does it make any sense? So the results. Well, first of all, the last piece to introduce was differentiation cost, and it seems to be necessary, so I start from there. So here is the screen of different differentiation costs, and the colors show what are the likelihood to get the reversible somatic differentiation. So the costs are on the axis. The maturity size is fixed, it's 10 cell generation, 1000 cells or so. And for every pixel on this map, we sampled 200 different random parameter sets for somaral benefits profile. So we screen for different shapes, find the optimal strategy for every one of those, and look what fraction of those sets promotes reversible differentiation. So you see that if the costs are 0, both, it's just 0. It doesn't evolve. But if we introduce cost in any direction, we have a reversible differentiation. That's nice. It's not that abundant. I'm talking about like 1 to 2% of the whole set providing reversible differentiation, but it's something. The greatest abundance of reversible differentiation is where it's very easy to turn into soma and very difficult to come back. But that's intuitive. That's the strongest effect, and it's a boring effect. It's easy to go into soma, and it doesn't worthwhile to come back. You just develop a reversible somatic differentiation and don't even try to go in opposite direction. But we find sometimes a reversible differentiation in the opposite scenario, when it's very difficult to go from germ to soma and very easy to come back, and still you go from germ to soma and never come back. And we mostly focus on the case on the main diagonal, where the costs are the same. Because even there, the irreversible somatic differentiation does not evolve because it's easier to go one direction than another.
Can I ask a question?
Yes, please.
For more clarification, in terms of a germ rule cost and a somatic rule cost, like, because in terms of mammalian cells differentiating, you start with what would be potentially, I don't know, a germ cell, like let's say a stem cell, and that has the capacity to be anything. And then it becomes a soma cell. But in your case, your germ cells are able to differentiate and your somatic cells are able to differentiate, and differentiating is costing them, like they're changing into something. I just want to know, what does this cost like mean in terms of,
well, they
like
do
every
different...
step they take into division? Is that what you mean?
Sorry?
Is it like every division step, or what is this cost? Just
yes, it's applied at
yeah.
The costs are the resources that you need to invest into the differentiation specifically. Like in addition to division, a cell has to do something else to make the offspring different.
Right.
And therefore, it needs to invest additional resources and additional time. And therefore, it slows down the whole life cycle, because those resources can be used in something else, into more growth, for example.
Oh, okay. Yeah.
So, and those costs they are applied at every time the cell divides.
Okay. Yeah. Thank you.
So yeah, the first finding, we need some costs. We need some penalty for differentiation to get into the irreversible differentiation land. So the next finding is that reversible differentiation needs quite large organisms. So what do we have here is we have cell differentiation costs in general. So now we assume that they are the same. Now we screen through the maturity size from just 2 cells, like you divide and immediately go, up to the power of 15. That's like 32,000 cell organism. That's almost macroscopic size. And the colors are the same. So for every pixel, we have a number of different patterns, and we want to know which of those patterns promote evolution of reversible differentiation. So we don't see anything on sizes from up to 2 to the 5. So 32 cell organism doesn't show us any irreversible differentiation. So there is a sort of soft limit. It is soft limit because if I make costs of differentiation different, I can get into this area, into 2 to the 5. But I need to have a costs really, really asymmetric. So like for 2 to the 5, I need like at least twice the cost in one direction than in another. So then we go into the profiles, because what remains are the profiles. So we now fix the cost. We fix the maturity sizes at 7, 15 generations. And what I show is the medians, the thick lines, and the 90% confidence intervals, that's those shaded areas. For the profiles, the gray, all the profiles that we have, and the colorful lines are the profiles that promote reversible differentiation. So what do we see here is a very fast decrease in the cell doubling time. So like, even if you have 20%, only 1/5 of your cells are somatic, you already have 5-time increase in the 5-time decrease in the doubling time. So some very, very efficient soma that even a few somatic cells already make a difference for the organism. That's where the irreversible somatic differentiation evolves. And the requirement is more strict for smaller organism at 7 than for the larger organism. Like if you have to divide at size 2 to the power 15, then you can relax this condition a little bit, but not that much. So again, looking at the shapes, so we have like 4 parameters. Yes, please.
Yeah, in the previous one, when you say the doubling time decreases, that means the time taken for the cells to divide decreases, or it increases?
The cells divides faster. Like somatic cells move organism so fast that it accumulates more resources, so it needs less time to divide again, to accumulate enough biomass to divide.
Oh, okay.
So the lower values here are the
outnumber,
better for the organism.
So as the number increases, somatic cells outnumber the germ cells.
Not necessarily. They increase in numbers in irreversible differentiation, but they may in principle do it very slowly. But
Okay.
if you wait for long enough, eventually, yes, majority of the organism will be mostly soma.
Because the x-axis is fraction of soma role cells, right? It's getting decreased. That's why I was asking, because they are a bit contrary to both. Because as the cell doubling time decreases, should it not mean more divisions and more cells accumulate much faster?
Yeah, so it starts with 0, it divides, it differentiates, and now we have some somatic cells, and they provide benefits. So the next division is faster. And in irreversible somatic differentiation, every next time you land in a more and more somatic cells, so it will go faster and faster.
Okay.
Until it reaches saturation. So somewhere here, it doesn't matter how much soma cells do you have. Does it answer your question?
Yes, thank you.
So looking at the parameters, those profiles, they're controlled by 4 parameters. It's hard to plot all of them together because it needs us to operate in 4-dimensional space. I will break it in 2 plots. So if we go for maximal benefit, so how much you can in principle get from the soma, and the contribution threshold, so how much soma do you need to have at least some benefit, we find that the profiles that promote irreversible differentiation, those green subset out of gray ones. So now it's 3000, not 200, it's an additional simulation. So what we find, we need a small contribution threshold. So the soma cells must contribute early, as we saw before. And the maximal benefit should be quite significant, but not too much. If we go to values close to 1, like instant division, that's mathematical abstraction, of course, but it's a theoretical one, then we again lose irreversible differentiation. But still, the final effect must be significant, at least half, as you see. So at least double speed of division comparing with no differentiation scenario. If we go for 2 other parameters, the saturation threshold and what we call synergy, it just explains what the shape is. It convex or concave? We find it's a situation of either or. Either you have very high synergy, so effectively you starting from the moment when soma cells start to contribute, the line just drops down, or it means it immediately gets a lot of benefit. And then it doesn't matter what is the saturation threshold, like in this area. Or you have very low saturation threshold, very close to the contribution. So the transition from maximum to minimum is very narrow. And then it doesn't matter what is the shape of that transition, because again, you immediately falls very much down to the bottom to get the maximal speed of development and continue from there on. So that's the results. And the big question of theoretical biology is always, do those results have any meaning? Because it's very easy to run the model, but does it make any sense? So from theoretical perspective, the most contradictory for me part of the model are those differentiation costs. Like we designed in a very specific way, We put the differentiation in a very specific way, and maybe we just study the artifact of the model. Why not? So what we did, okay, to be honest, it was not us, it was a reviewer who said that. Great credit to our reviewers. Thank you to them. We did alternative model. We, instead of costly differentiation, we went for a risky differentiation. So division times is always the same. However you divide, it always you don't have any penalty for differentiation. Not that they are all the same. We have still contribution factor. It's a bad wording from my side, sorry. But every differentiation events create a risk that there's something will go wrong, that maybe there will be some selfish cell arise. Think of a cancer. And that cell will just take over the organism and kill the organism at the end of the day. So there are risks that accumulate during the life cycles, and the more differentiation you get, the higher are the risks. And then at the end of life cycle, you either check, are you fine? If yes, then you go for reproduction, all somatic cells dies, everything happens as usual. If not, if you're unlucky, then the entire organism dies, no offspring whatsoever. And then we repeated everything, not everything, but the first part of our simulation. And they screen for the costs, and now it's not the cost, but risks factor. And we see essentially the same. We see that the reversible somatic differentiation doesn't evolve at no risk, but if you introduce any risk into any direction, you get the irreversible differentiation, and the maps are qualitatively the same. So we have an area at the top left, and now it's a bigger area, but still. So we think that this idea of the penalty associated with cell differentiation, it makes a difference for emergence of soma cells and a classical understanding of it. And then even more complex question, are our results biologically meaningful? So our first finding, we need costs. Okay, we know that there are costs. So we know that once cell differentiate, protein composition changes, and somebody has to pay for it, and it's organism itself, of course. So if this organism didn't do the cell differentiation in cyanobacteria, so those data for cyanobacteria, then it could grow faster. It delegates those resources to the growth instead of differentiation, but still they produce those heterocysts. But to be honest, I have to admit that differentiation costs, it's something that is unavoidable. You cannot run a control experiment for that. I cannot imagine how one can do it. How can you turn off the costs of differentiation? So probably that particular finding is valuable theoretically for our thinking, but it's not something that can be tested experimentally. Then we go for our second finding, and the organism size criteria. And here we go into the volvocalis groups of organisms. That's a monophyletic group of organisms. That's a green algae. They are of different sizes. And the largest one are clearly multicellular organism with a lot of complex stuff going on. And the simplest one are just single cell organisms. And what we find is that the very first hints of differentiation happens in the organism called Eudarina. It is 2 to the 5, 2 to the 6 cells. So typically it's 32. And out of those 32 cells, the 4 of those are a little bit smaller. And they have somewhat lower chance to give rise to a new colony of the organisms. And then in Pandarina, it's size to the 2 to the 7. We have clearly germ-soma differentiation, and Volvox keeps the trend and promotes it even further. But that's evolution of complexity. So far, it looks at least promising that this threshold for the size really makes sense for the organisms. And finally, that the somatic cells should be effective in small numbers. Our profiles, they drop down very quickly. So even if you have few cells, you have a lot of benefits. Again, as I said, Eudarina, in this particular species, it does produce soma, and it does produce it in a very small number. Okay, maybe not truly soma, but something very close to specialization. and it just make 4 cells into there. So something significant happens within the first few cells. So it keeps producing them over and over for millions of years. I don't know how much is the benefit at the end of the day, but the threshold is certainly quite low. So I would like to create my team. So most of the, well, all heavy lifting here was done by Dr. Yuanxiao Gao, who soon will be the lecturer in Shaanxi University in Science and Technology. Just last year, she defended her PhD and now she moves on. That's great, she's fantastic. We have Hejin Park, who is now a professor. At the time, she was a postdoc, now a professor in Han University in South Korea. And our big boss, Arne Traulsen, who is here. Oh, he's great. Yeah, and thank you for coming. And there is a paper in eLife with more details if you wish to, or if I cannot answer it to you here, you probably can find answers there. Thank you very much.
Thank you, Yuri. This was great. I also posted the link to the papers in the chat for anybody who wants to go there.
Thank you.
I can start with questions. Well, if so, I know you showed a lot of this in terms of the bacteria world, but when you were talking about maturity size, how did that work into your model? Was there a cost, or was there some thing that the cells had to achieve? Like, what if all these cells had to work to achieve a particular size, as in mammals? Like, we have to get to an 8-cell stage, a 32-cell stage. I don't know if there's something directing them. And I wonder, is there a cost to, or some sort of a risk factor of not achieving a right size that can play a role in these decisions? Does that make sense?
Yep. Well, so far the model is very minimal on that aspect. We assume a perfect phenotype control. So like they can always achieve there. And there is no cost associated with specifically getting there and reproducing exactly at that space. But it would be interesting to study what happens if you have a variety in the maturity sizes. So I agree that it's a great question, but I cannot answer it, unfortunately, today.
Aditya, you can go.
Yeah, hi, nice talk. While listening to your last part in this slide, I had this hunch that while we are dealing with human embryos, only after the 32 stage, the differentiation starts. So does this model have any specific thing? Because like that was the biological observation. So is it something related in that way?
Wow, that's awesome. That's awesome. I'm really glad to hear that. Yes, it formally fits, but I have to admit that the model has nothing to do with human embryos, because the human life cycle, it just started at 32 cells. And we are thinking of the guys that must compete with all the predators and everything while being so small. But maybe it's, well, this threshold is so nice that evolution keep it forever, and at least in our lineage.
It starts with a single cell, and as it goes, it progresses to multiple evolutionary stages before it becoming human. So just had a hunch whether does this model have that. That was the first one. And second one, when you are saying about somatic differentiation, as the organism evolves, multiple different types of somatic cells come in. So are you trying to incorporate any bias towards different somatic differentiations rather than generally saying that one somatic differentiation is enough? And the number of germ cells which have in an organism are very much limited. So is the cost which you are making specific to one or the other?
Well, for multiple somatic cell types, it would be really interesting to get there, and that is essentially a plan how to develop this model further. But like with 2 cell types, things become more complicated and it needs thinking. and it needs a model design. So I cannot talk about it right now. There is nothing, no results. As for the germ cell control, if I understood your question correctly, again, there is no control for the number of cells by the end of the day. We allow it to be emergent. So there are some rules in the system. We let it go. We see what comes at the end of the day. Since the strategy itself is stochastic, I don't think we have really control for the cell numbers. So different organisms are really different in here. So they follow different trajectories and may end up with very diverse composition.
On this slide, maybe I can ask a naive question.
Please.
You require that the organism should be large enough. Is this in the sense of critical mass for a process to happen?
It may look like critical mass, but my understanding of it, that I didn't test, just intuition, is that it needs a number of steps. Like you need to have at least 5 steps of cell division and proper differentiation to extract some value from the irreversible differentiation. Because if you have only 3 steps available, you have to produce your soma cells at a very high rate, and then to make it matter during the life cycle. And then you have very little soma at the end of the day. Very little germ cells, because you have to produce soma cells very fast. And if you are allowed to grow up to, say, size of 1,000 or at least 100 cells, then you can make those processes more gradual. It's less discrete. You can allow for fine tuning of the numbers of germ and soma. And that's where the irreversible differentiation gets a chance. But I don't think it's about the end state, because the final state.
How about the first point? Like this is number 2. There is a first point. Can you show this slide again? Number 1.
Wait.
Number 1.
Number 1 here.
Yeah. So if changing the role is costly, now there was something like costs are necessary to evolve differentiation, for evolution differentiation.
Yep.
You had another point like that, or you said it. So I don't know, that reminds me something very strange from a different field about the costs that are, or work of proof that people require in Bitcoin. So I don't know if it's this too funny anecdotal connection here, but anyway.
But this is the cost of maintenance. I have
but if cost is necessary for something to happen, this is like, it tells me the nature of this process. It's a restriction of what kind of process can take place.
Oh, yes, sure.
So it's interesting. Anyway, I don't know how to comment on the third point, because the irreversible is a bit tricky part of all this talk. So irreversible differentiation. So maybe we can dissect it. What is reversible differentiation?
Reversible for us is a situation, wait a moment. Yep, when you can go back and forth between cell types, between cell roles. Germ, next division is soma, next division is germ role again.
Okay,
that's
sorry,
reversible.
this is clear. I just missed this point. Thank you.
Sorry for being very fast there.
No, you were fine. You were absolutely great.
So I have another question regarding the costs. It seems like a pretty popular topic. But if the model, for example, or if the function deviates from the model, does that mean that there was a change in the composition of within the cells or outside the cells? Since I think the costs are related to the properties the cell has or that it needs to grow.
I didn't get your question. What do you mean function deviates from the model?
So I think it's the graphs of growth. Yeah, that one.
Mhm.
So if we see any changes in the time, or yeah, in the doubling time, is it only depending on the fraction of soma roll cells, or does it also depend on the composition of the soma roll cells?
At that stage it's only the fraction of the soma roll cells. So you may think of the whole process as you need an amount of resources to accumulate, to go from one state to
Yes.
another. And how much resources do you need depends on how much differentiation do you want to get. If you have no differentiation plant, then you need a little bit of resources just for cell division. If you want more differentiation, then you need to accumulate more and more resources to get from here to there.
Okay.
And the number of soma cells now define how fast those resources will be accumulated. So overall, the time necessary depends on both, on the composition that you have here and the composition you want to get there.
So the resources the cell have, it's seen as the fraction of soma roll cells, or can it be accounted as a different parameter?
In principle, this model can be formulated with a separated parameter for the resources.
Okay.
But for just streamlining the mathematical work, we just have doubling times and assumptions.
Okay. So another question is if during this, is the time attributed in the model as the cycle of the cells?
The time of what? Of the whole cell life cycle?
Yes, because I think you mentioned at the beginning that tasks of the cell are distributed in time. So how is that time seen in the function as the cell cycle?
We don't have a cell cycle, like changes of the cell stage during the life cycle of the whole organism.
Yeah.
So we have a life cycle that consists of multiple steps. Like in here, we have 3 steps. And between steps, the composition of the organism changes. So it starts with only germ, it accumulates some soma, this fraction can go up and down. And between those steps in the life cycle, the composition changes. And the cell that was a soma-like cell in here, it can give rise to germ-like cell there because of the differentiation. And then I consider that this is a scenario of reversible differentiation because soma comes back into germ potentially.
Okay. So can the germ roll cell differentiation and the soma roll cell differentiation depend on each other? So the amount of germ roll cells and the soma roll cells be like in the same function or graph, since both can depend on the same resources, and maybe one consumes more of a resource than the other?
Yep. So this question has many aspects. So I will break into a few sub-answers. So
Okay.
first of all, the differentiation program can change with time. So we assume that the probabilities to differentiate are all the same independently on your stage.
Okay.
It is in principle possible to design a model where they change. So like differentiation for that guy is different than differentiation if it's here. But we don't do it in this model. In our case, it's just constant. Another thing is that we assume that cell divides simultaneously. So whatever resources they need, each of those cells, we just pull them together and wait until all cells have enough resources to do whatever they want, and then they all divide. It is possible to design a model, and we try it even, design a model where each cell has its own resource pool, and they divide asynchronously. And we have a little bit of that in the paper, but those models are extremely demanding computationally. I mean, even what I present to you, it's weeks and weeks of simulations. And when cell divides independently and we go deep into the details, it's just unfeasible to do the analysis that we perform here. But we did a little in the paper. We have something in appendix.
Okay. Thank you so much.
Thank you.
I have one more question. And this is more about like did you see a system where you started really fast, let's say doubling time, and that doubling time then slowed down?
We didn't look specifically for that, but I guess, well, in
You had the
reversible,
opposite, right?
you can do it.
But I meant you had the opposite. It looked like everything started from 1, which was slow doubling time. And
Yes,
then
they have to start.
And things went faster. Is that correct?
Yep.
Okay. The reason I ask is typically in mammalian systems, and this might be something to address later, they start fast. So like the early embryo is seen to divide much faster. And then as cells differentiate and change, doubling time is actually slower. And that would be a little more applicable, I guess, scenario. And I was just curious to see if that.
Well, my naive feeling, just because I never considered this model in the context of mammalian embryos.
Right, I know. We're just pushing you here. I don't mean to do that.
Yes.
I just meant we're just pushing you out of your limits, but it'd just be cool to think about it.
Yeah, so after like 5 minutes of thinking about it, probably this is achieved by other processes that I consider in the model and for other reasons that I have in the model. So it's not about optimization of the embryo growth rate, like for sure. I'm a physicist. I can say, yeah, it's not about that. But it would be interesting to think about it. Why do they do so?
Yeah, I'd be happy to debate this longer with you. So
yeah,
definitely, I'll bug you.
Maybe one more question from me, not about complex multicellular organisms, staying with these simple ones. I'm super naive to them. So you assume that these 6 parameters, right? You assume that each of the 2 types, you had that slide with the 6 parameters.
That one, like.
Yeah, during the division, type A goes to A or B with this rate, and so on. Is this just based on like the phenomenology of these simple multicellular organisms that they can't differentiate without dividing based on observations and that?
No. That was, to be honest, just the simplest way I can set up it into the model. So okay. I link differentiation with the cell division. It's probably what happens in the more complex organisms, at least sometimes, let's say. I'm not sure that it is what happens in the simple organisms. And I use probabilities, again, as the simplest way of implementation.
Okay. Yeah, I asked because in the context of like mammalian cells, I guess, certain cells have kind of progenitor roles where they divide very rapidly, like produce many, many cells, and where others don't divide at all, which doesn't totally align with this picture for the simple ones. If you see what I'm saying.
Yep. So different cells do have different division rates, but at least in smaller organisms, we quite occasionally find synchronous cell divisions, especially in the simple and small ones, because, well, maybe it's a trait that is not preserved until the larger and more complex species, but that's where it started. And this model is tailored so far to very first steps into the evolution of complexity.
Got it. Thanks.
It's really interesting, right? Like the fact that you see very little evolution of irreversible. And I think, Matt, you started this by saying, you know, we're able to take a skin cell and reverse it back to something. And so that might be an interesting like aspect where you can say, you know, there's a higher probability that things are most likely reversible, but something else is forcing them to maintain some state. I don't know, maybe it becomes like the environment or something else. But your theory would then hold that majority of our cells can potentially be reversible. It's just something else is keeping them. And this irreversibility or the lack of irreversibility is a unifying principle.
Yep.
If it makes sense. Are there any other questions? No. Well, thank you, Yuri. This was.
Thank you very much for inviting me, Maria. Thank you for coming.
Thank you all for
Thanks.
being here. I hope if there are any other questions, just you can message Yuri or I, and we'll get back to you.
I'm happy to chat and discuss.
Oh, I'll be bugging you.
Yeah.
Right. Bye, guys. Bye, everyone.
Bye-bye.
Bye.
Thank you very much, Yuri.
Thank you. It was very nice.
No, it was great.
I like the questions. I'm not sure that I was able to answer